Thank you all for your patience. Thank you for coming. So today we are going to be talking about the group time tree. So this is an expansion of the FTDNA Discover tool that we came out with. And that in turn is an expansion of the haplotree. So in order to understand the group tree, I think it's important first to understand how the time tree works, and then also to understand how the haplotree works. And I'm the sort of person who really likes to understand the science behind something, because that gives me a better understanding of the information that I'm looking at and what it means for me. So first, let's go ahead and talk a little bit about haplogroups. So haplogroups are basically migration routes out of Africa. And the oldest humans that we know of currently come from Eastern Africa. And this is tens of thousands of years ago. And from there, they spread all across the globe. And as the different groups of people separated one from another, they started to develop their own unique mutations. Now, these mutations, they're just small changes in the DNA. So DNA is composed of 4 chemicals that we abbreviate as A, C, T, and G. And so a mutation is just where in a particular spot, instead of an A, there happens to be a T or a C or something like that. And in most cases, it doesn't affect how the body functions or your health or things like that, but it does help to create all of the diversity that we see today. And it's especially important for genealogists because these different groups of people started developing their own mutations. And so we were able to genetically tell one group of people apart from another based on the mutations that they have. So the type of mutations that we use for the haplotree are called SNPs, or single nucleotide polymorphisms. So what does that mean? It means that it's in a single location on the chromosome. So you're not talking about a string of mutations. It's just one single mutation in one location. And the polymorphism just means that it has changed its value from A to T or something like that. So we have all of these different groups of people, and they each have their own unique sets of mutations. And we're able to see in present day test takers that some of them are the same, some of them are different. One group of people might overlap with another, but not a third. So how are we able to determine how all of these mutations came about? How are we able to make a time frame of it? If you look at the map here, how, for example, are we able to tell that there was a branch that happened here and not somewhere else? Well, the way that we do that is basically by process of elimination. So for an example, let's take a really simplified example. Let's say that this is an individual who has 3 specific SNPs. So we'll just call them A, B, and C. Let's say that person has 3 descendants. And those 3 descendants, they all have A, B, and C, but one of those people happen to have D. Well, we're able to tell then that D came about after A, B, and C, because the parent had it, but not the child. And even if we don't have the parent there, we're able to tell based on the similarities that they have that D is older. Even if we had only the children of that, we could see that they all share A, B, and C in common, but only one of them has D. So that one's most likely going to be the older one. So then we highlight this. And then let's say this person goes on to have 3 descendants of their own. They pass on A, B, C, and D. And then they have one descendant that passes on here, this extra E. Now, we know that these A, B, C, D people are descendants of A, B, C, D and not A, B, C, D, not A, B, C, because they have that extra D. And I'm sorry, that's my cat. She likes to participate. So here we have then E, again, through process of elimination, we know that E is younger than A, B, C, and D. So this is a really simplified example because we're talking about hundreds, if not thousands of people. This is Chi-Chi, by the way. Chi-Chi says hi. So the very first haplotree that we came out with, or well, it wasn't actually us that came out with it. It was the Y chromosome consortium, which was led by Michael Hammer. And this came out, and you can see here that basically it's the exact tree in our example where we have all of these different branches that are all coming off of one another. And this started growing more and more and more. This was the first one. You can see that there's, a few dozen, maybe 100 SNPs that are on it. Today, there are hundreds of thousands of SNPs, and Big Y has really helped to expand that. So to make it a little bit easier to understand what we're looking at, a few years back, we developed the block tree. So the block tree, it is essentially telling you the same thing that the haplotree is, but it's grouping these SNPs together. So in each block, you can see that there's multiple SNPs. And what that means is that we don't know if those SNPs are older or younger than other SNPs, because so far we have like everyone that we've come across that has that E SNP shares everyone else who also has the D SNP. So we don't know if D or E is older. It takes another person coming along who doesn't have E for us to determine that E is in fact the younger one. Oh my goodness. I'm so sorry. She was sick. She had this nasty cold for like the past week, and she finally got over it. And so now she needs to catch up on not feeling good. So I am sorry about that. So these SNPs, they're grouped together by essentially what are called equivalencies. We don't know if they're younger or if they're older, but we do know, for example, that this block is older than this block. So over here on the side, you can see these numbers. These represent generations. So we typically say that a generation is about 25 years. It can vary, obviously. But it means that this block is probably going to be about 10 years old. Now, it can be a little bit hazy, though, because different blocks might have a different number of SNPs in them. So it can be a little bit misleading if one block has a ton of equivalencies, because it might look like it stretches across more generations than it actually does. So what we did was we developed the time tree. We basically took all of that same information, but instead of blocks, we turned it into a linear graph here. And let's see. I put the slides out of order, but here's a little bit of a blow up of it. So you can see here that there's this SNP. It gives rise to these individual branches. So this is able to give you a better lineage or a better view of the different lineages. The countries that are listed over here at the end, these are self-reported. So these are test takers who have tested positive for those SNPs, who have reported their most distant known ancestor as being from a particular country. So as genealogists know, you always have to take that kind of information with a bit of grain of salt. But even if one of the people or a handful of the people were incorrect in their assessment, you can still see that overwhelmingly it's the same. So it's a pretty good indication that that's where you're supposed to be looking at. So from the Discover tool, if you go to just FamilyTreeDNA or discover.familytreedna.com, it'll take you to this page where you can enter any haplotype.
Jim, real quick, we can't hear you.
Can you hear me now?
We can.
Okay.
Go ahead and
All right.
start over on this. Go back like a couple sentences.
Okay. So again, sorry about that. Apparently today is just not my day. So all of these are self-reported countries. So even if some of them are incorrect because people didn't do their correct genealogy or anything like that, you can still see that overwhelmingly it's the same. So that gives you a good indication. So these are really helpful for SNPs, but what if you're trying to find a specific lineage within your group? Like, for example, my last name is Brewster, and through my own genealogy, I know that I'm a descendant of William Brewster from the Mayflower, but how do I know that? Say another Brewster comes along and I want to know, well, are they from William Brewster or are they from some other line? Well, if I know what SNP I'm looking for, I can see this, but it can get a little bit hazy. What if I want to just look at all the other Brewsters and see how they all compare? Well, that's why we went ahead and we implemented it to group projects as well. So from this same the Discover tool, if you click here where it has a haplogroup, you can instead select group project. And if that group project has made the results publicly available, then you'll be able to see everyone who is in the project. Because of privacy reasons, there's some projects who choose to make it private and you're not going to be able to see those. But in this example here are all of the people that are in this particular project. So this is the McAllister project, which I'm an admin of. And so in addition to countries over here, you're also able to see the name of their most distant known ancestor and any information that they put along with that, such as a birth date or a death date. So here we've selected all search results. So this is going to show everyone who is in the group project. But as you can see, that is going to be a lot of people and a lot of lineages, and this is only part of it. This, you can scroll down further and further and further and see more and more people, and you can see that the time frame is truncated because the ancestral SNP is thousands of years old, whereas all of the different branches are hundreds of years old. So it's oftentimes more advantageous to select a specific subgroup. So in this example, we've selected here this subgroup and all of that ancestral information that goes along with it. So this is able to tell you that here within this subgroup, here is one distinct lineage, here is another distinct lineage, here is a third distinct lineage. So this gives you a better idea of how you might be related to someone else in the project. You can also see here that there are these dotted lines with which are the time frame that we estimate. So this SNP we estimate is at this age, but it could fall anywhere within here. So you might be asking, how do we determine the age of them? Because we've determined their relative order. We know that one SNP is older than another one, but how do they fit together in the, in terms of years? Well, we're able to do that in most cases by comparing archaeological remains to living day people. So if you have a burial site and you're able to date the age of that burial either through carbon dating of the bones or dating the age of the soil where they're buried or the age of the artifacts, the pottery that's in them, there's a number of different methods. But if you're able to determine the age of that, of those remains, then you get a pretty good idea of what time frame you're looking at. So you can take the group of SNPs that you find in that archaeological sample and then compare it to other descendants who are in the area, see what they have in common, see what they have different. So if you have, say, 50 SNPs in the present day population that you don't see in the archaeological remains, then you're able to tell, okay, well, those 50 SNPs then occurred in the time frame. Say this sample is 6,000 years old. Well, those 50 SNPs occurred in that 6,000 year period. So you're able to divide 6,000 by 50, get an average rate of how many years or how many generations between each of the mutations. Now, this is variable depending on the population and the number of available dig sites and test takers that we have today. So that does get taken into account. It also includes things like those equivalent SNPs, ones that we don't know are older or younger. So there's a lot of different factors that go into determining this age. We also try to verify the age of them by comparing the SNPs to the known genealogies, the proven genealogies, to see if those numbers line up. When we first released the beta version, we saw that some of the estimates were a little too young. So we went back, we changed some of the algorithms. We got a better idea that seems to line up pretty well with most people's genealogies. We are always improving it. Some lines are more documented than others, and some are better represented in our database, so it's able to refine those dates a little bit better. So this here, for example, this SNP, it is based on all of the different factors that I mentioned. It could fall anywhere here within this time frame, and this is the mean time that it is. So it's the most average, it's the most probable. It could fall somewhere in here. You may also be thinking, okay, well, I have my genealogy, it doesn't match into the time frame that you guys are listing. If that happens, let us know, because we are always working on improving it. And then also keep in mind that this is an estimate. So like anything else, it's best to take with a grain of salt. So one other thing that I want to mention here is, like I said, this is for public projects only. And it's also if the individuals have made their information publicly available. So this person here, you see, has the lock icon. It means that they haven't made that information available, so it's not going to be there. Also, this information is based on SNP test takers only. So if a person has an STR test with a predicted date instead of a confirmed date that's confirmed through SNPs, it's not going to appear here. They would need to get SNP testing like the big Y to be able to confirm that. So that's pretty much all that I have prepared. I wanted to leave a lot of time for questions and answers, because I'm guessing the audience varies a lot in experience level. So I don't want to overwhelm some people, but I don't want to bore others. So I just wanted to open up the rest of the time for Q&A. So if I don't have a chance to address your question here during this presentation, feel free to stop by the booth. I'm happy to help. And then also feel free to give us a call or send us an email to our contact center, and we'll be happy to help there too. So that is the end. Yay.
Hey, Jim.
Hooray.
So we do have quite a few questions that are coming in. Do you mind if I read them off to you and you can answer them here?
Yeah, that would be
Awesome.
best. I know it's been coming in.
Yeah, there's been a few. You guys, Jim is the expert. I'm just here to assist. So there's been a few that I've been able to answer, but most of these are for you. So the first one is from Jenny Sharp. She's wondering, what about X haplogroups?
Can you, Jenny, can you clarify what you mean by an X haplogroup?
Go ahead, Jenny. If you want to just submit back into the questions and answers, we'll see your follow up there. While she's typing that, we'll go to the next one. And the group time tree, how do we know how to find our matches in the subgroup pull down on the left?
Because of privacy concerns, you're not able to see matches, the name of matches, because this is publicly available information, so it doesn't have names associated with it. You are able to see subgroups. So if you're a member of a group project, when you go to the STR results page, you can see the subgroups that are there, and whatever subgroup you're in, it lists the other kit numbers and it lists the most distant known ancestor, but it doesn't give the name of the matches. When you go to your match list through your dashboard, you can filter matches to only those who are in a specific group project with you. So you're able to then pinpoint out who those people are and contact them. But you would need to contact them directly or the group administrator to see if they the names behind those dates. Which is unfortunate, but also it's a privacy concern.
Awesome. Thank you, Jim. Our next one comes from the Mesa, Arizona Alma Steak. They're asking, how big is a SNP?
So a SNP is actually it's pretty small. It is just like I said, it is in a single location. Let me see here if I can pull something up. Give me just a moment here.
No worries.
All right, hang on a second. I just want to get a visual for you that I think will help you out. All right, here we go. Okay, this is a slide from my presentation that's available through our booth, which is the fault in our STRs. I'm pretty proud of that title. It's pretty punny. So you can see here, these are STRs. So these are short tandem repeats. So these, in this example, the STR, it covers all of these locations on the chromosome. A SNP, on the other hand, is only going to be in a single location. So it's only going to be this C or this T or this G. And the reason that we base the results on SNPs and not STRs is the chances of just this one little location changing is a lot lower than the chances that anything here might change. Once a SNP occurs, once a mutation occurs, it's very unlikely to change back. So it's a lot more stable. And it can help pinpoint more accurately in time where it is rather than STRs. So more answer than you needed, but the answer is it's just one single nucleotide. Can everyone hear me?
I'm muted. That was the problem. The next question, I can't pronounce his name and I don't want to butcher it, but they're asking, you mentioned that if we have genealogy information that could inform the time estimation, we should let you know. How can I do that?
So with that, the best thing to do would be to email us, and that's going to be at groups@ftdna.com.
Perfect. Next question, does it work if only one person in a surname group has ordered big Y, or do lots of people in the surname group need to order Big Y in order to get good results?
The more people who test Big Y is going to dramatically increase the information that you can get out of it. This isn't necessarily strictly Big Y test takers, because there are some other SNP tests. For example, National Geographic Project, they did the Genographic Project, which was a SNP-based test, and people could transfer those results. If they did, then those SNPs would also be listed here, but primarily the data is coming from Big Y. So the more people who test, definitely it helps out with that.
Awesome. Let's see this next question. It looks like the sound went out, right? Excuse me, when you were talking about what is the URL to get to the search screen?
Oh, for the search screen, it's discover.familytreedna.com.
Awesome, thank you.
And also, you can find it if you just do a Google search for FTDNA Discover. It'll take you right there.
Okay, cool. Okay, so the next question, sorry, my son just ran in. We have a couple questions, multiple questions about how do group admins use the time tree?
So the subgroups that are listed in the time tree are created by the group admins. So if you are an admin, if you're familiar with GAP, there's a section for member subgrouping, and that's where you can create subgroups, move members from one to another, and that information is what's going to be displayed on the Discover time tree. So that's it does two really important things. One is it's able to give you a better visualization of this subgroup, because if you're familiar with the colorized chart or the classic chart, the STRs are just a long string of numbers, and it can be somewhat difficult to intuit what all those numbers mean. The other thing that it can do is it uses SNPs instead of STRs. So like the example that I showed with the different lineages, it can help identify lineages within subgroups in a way that STRs can't. In terms of how the admin uses it, it's also the public tool that they go to. So they would be seeing the same information that the group members or the public would. The only difference is that they're the ones in charge of the subgrouping.
Okay. We have one here. How is this going to compare to the YFull age estimate tools?
So YFull, I personally don't use YFull, but from what I understand, okay, so with the SNPs that we report, we need to get at least a 10x confidence rating. So let me explain what that means. The test is the Big Y is an exploratory test. So it looks at a SNP rich region of the Y chromosome. It picks up all of the SNPs that it can. It runs that same region over and over and over up to 70 times. And that way we can make sure that there wasn't some sort of misread, that we thought we picked up a mutation, but really we didn't. And so the threshold that we have to report those SNPs is 10. Anything less than that, and we're not confident that it's correct, and we want to make sure that the results we're giving are correct. YFull, from what I understand, doesn't have that same limit because all of the reads from all of those 70x reads, that's available in the BAM file that you can download. And when you upload it to YFull, they pick up all the positive reads regardless of the confidence level of them. So in some cases that's a bad thing. In some cases it's a good thing. It's really up to opinion, because there are some cases where Michael Sager, our Y haplotree specialist, he'll go in, and even if there's fewer than 10 reads, he can tell that it's quality. It's probably a accurate call, and that's due to another set of factors that I won't go into. But WIFL, you could get different informations. As to the tools itself, like I said, I'm not familiar with the WIFL tools, so I can't speak to that. But that's why you might see a difference in reporting of the SNPs.
All right. Next question, where do you primarily get your ancient DNA samples from, and do you get any associated archaeological data with those ancient DNA samples?
Well, so the ancient DNA samples come primarily from archaeological digs, and those come from scholarly publications in regards to them. I went to school for anthropology, so my bachelor degree in anthropology pretty much makes me the top expert in the field. So I can confidently tell you that usually when you find a burial, there are other artifacts that are associated with it. You find grave goods, you find usually settlements, artifacts, things like that. So that all tells its own set of information as well. One really good example is there was a case study not a few months ago that I read where you had a group of people who were buried together that were apart. Like, it was a graveyard, and there was one small section that was sort of apart from the rest of it that had a mixture of different genders that were in it, both male and female, whereas it was sort of segregated in other parts of the burial ground. And when they did DNA testing on it, they were able to determine that that was a family group. And really interestingly, there were two young male skeletons that were found that were identified as brothers. And then they did a mitochondrial analysis, and there were some female remains right next to it that were a mitochondrial match to them. So we're able to infer from that that was their mother. So that can tell you some really neat information about the site itself and customs, burial practices, family units in ancient cultures.
Awesome. Going through, y'all have a lot of questions. So I'm going through and looking at them. The next one, if you want to pull up the screen again, Jim, they're wondering if you can show the slide again that has the block tree with the red lines that match the data to the time tree.
Yes, let's go ahead. And that one, unfortunately, is a little bit fuzzy. That one I copied from the blog that we have. So let me go back to it. Oh.
While you're doing that, because I think this one might be pretty easy for you to answer. How long is a DNA sample, how long does it remain in good enough condition to perform a big Y test?
Confidently 2 years. But that's not to say that much older samples can't also get results. And that is true to one degree or another for all types of tests. And there's a lot of reasons behind that. First of all is the quality of sample. Were they just eating a cheeseburger right before they took the test, and there were like little bits of cheese in the vial, and we get some weird stuff sometimes. Well, that's obviously not going to be as high a quality of sample. The other factor is that when you submit a sample, you submit 2 vials. The first vial goes to the lab and it undergoes an extraction process. Basically, what it does is it pulls out the DNA, gets rid of like the cell membranes, all of the other junk that's floating around in it, bacteria, things like that. Just to run the DNA. But the problem is that all those cell membranes and that other stuff, it helps preserve the DNA, which means an extracted sample degrades more quickly than a sample in storage does. So that 2 years is pretty much for an extracted sample. With a sample in storage, it can be much, much longer than that. I've seen cases of samples that are 15 years old. getting big Y results. It really varies because of those reasons. If you are concerned about the age of the sample and whether you're going to get results, then before you test, I would highly recommend contacting customer service. They'll be able to tell you how much sample you have in the system, how old it is, whether it's been extracted or not.
Okay.
So I do have the block tree up.
Okay.
And okay, so here we go. All right, so you can see here that each one of these blocks, it sort of gets, I guess you could say, reduced to a single SNP. And that's just a limitation of the system. Is whenever you have an equivalency of different SNPs, one of them is chosen for the lead SNP. What's called the lead SNP. It's the one that's represented. There's a number of different criteria that go into how the lead SNP is picked. One is if it's already a well-established SNP that hasn't been newly discovered in big Y. That one usually gets displayed. There's a lot of other things for it, but you can see here that, let me pull up my handy dandy laser pointer. So you can see all of these ones here, they get reduced just down to this one. Because this is considered to be the parent branch of these other ones. It's possible that the other equivalencies are parents to it as well. That's something that you have to check in the block tree, just to double check if you're curious.
Okay. Let's see, where was my next one here?
Yeah. And I'm able to see it here. So
Okay.
The question from Heather, let's see. My brother did the big Y test, and we have a cousin that tested Y37. It's estimated that our common ancestor is from approximately 1750. If the cousin upgrades to the big Y, will we get a better estimate of when our common ancestor occurred? Absolutely. Again, because that SNP is going to pinpoint it. The next question, what about the T haplogroup? That one I am not super familiar with. I know it's much more rare in our database than some of the other haplogroups. I would highly recommend checking out a T haplogroup project. They're going to be the experts in that could give you much better information than I could. The next one, please close with your kitty in the camera view. I think she got kind of a little miffed that I kept pushing her out of my lap. She's a very opinionated cat. And so she's probably all sulking somewhere right now. So I'm sorry. Let's see. I have no living male relatives. I've tried back in my mother's at line into 1600s. My haplogroup is H, very common. Can I find my earliest US mother? And looking for records, one was that she was born in Holland. Can I trace my H back further than such records? You're talking here about mitochondrial DNA. We are working on building a time tree for mitochondria through part of the Million Mito project. So you're right, H is very common. The problem with mitochondria is that it's very slow mutating. So like Y-DNA can go hundreds of years without changing. mtDNA can, go a lot longer than that without changing. So it definitely has its advantages, because if you're a mitochondrial match, you know exactly what line it came from. But the downside is that the match could be really far back in time. So I always say personally that mitochondria lasts longer because, as they say, a man's work is sun to sun, a woman's work is never done. So it works twice as hard. All right, let's see. I have no living male relatives. I'm sorry, that's the one that just read. Okay, let's see. Testers list their earliest known ancestor, but not the ancestor where they connect with another match. It would be interesting to know the dates and places at the different nodes where testees connect. I have examples of where two fourth cousins connect, but there is no way to include who that ancestor was. Their earliest known ancestor was 100 years earlier. If you have a specific node that you're looking for, I would do a search for that node. As to the specific information, that's actually a good question because I don't think it shows that. And I'm not sure what the rationale behind that is, but I will definitely pass that along. If you want also, when this is done, send me a direct message and we can chat some more about it. All right, let's see the next one. I made an error. My earliest UK mother in Almondbury, Yorkshire, her marriage in 1580. Can I go back further with H?
I think that was in regards to an earlier question.
Okay, yeah. Again, it sounds like there's probably more to the story there. If you want, send me, like I said, send me a direct message after this and we can chat some more about it, see if I can get a better answer for you. All right, let's see. I still don't understand how I would use this information to apply to my family tree. Well, you're able to, it's more along the lines of if you have a question about where a match connected to you from. If you're talking about your family tree, like the family tree that you've built through your paper trail genealogy, if you have known genealogy, known records, it's probably of your direct line. It's probably not going to help you out a lot. The biggest advantage of this is in finding connections within groups and within matches and seeing where common ancestors split apart. I hope that answers your question. The next one, new information for haplogroup E, BY 4526. That, again, I'm not an expert in the E haplogroup. That would be a good question for an E haplogroup project administrator. I know there's several E haplogroup projects. I'm not sure which that one falls under. There are two different ways that SNPs are identified. One is the shorthand, one is the longhand. When haplogroups first started to be discovered, they came out with a string of letters and numbers. So it's like, for example, you had the R haplogroup, and then you started finding separations within the R haplogroup. And so then you had R1 and R2. And, then within R2, you found more separations. You found, R2A and R2B. And so then you started getting like some just really long stretches of it, like R2, A2, 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 B2, B2. And by the time it got to R2, D2, we just realized that we needed a better system. So instead, we switched to just calling it R, followed by the SNP that identifies that haplogroup. So with and so I mention all of that because when you're searching for the E haplogroup, you might find that some of the haplogroups are listed as the E1 project or the E1B project. I know that's a pretty big one. So if you're not sure what subgroup is best for you, you can always do a search for haplogroup shorthand to longhand converter, put in what the SNP is, and then that will tell you what the longhand name of that haplogroup is, and you can see if there's a project that lists that. Hopefully that helps a little bit. All right, so the next one, is there any value in someone doing SNP testing if their match list is very small? Well, you can get big Y matches that you wouldn't get through Y STR testing. The thing with STR testing is that there are thresholds for every level. So my boss, actually, Janine Cloud, she has a really interesting case where she has hardly any matches for her uncle at the Y37 level, but she has even more matches at the Y111 level because they were just past the cutoff threshold for the 37 marker level, but not the 111. So doing that additional testing opened it up. So if you're asking, is there value in testing? Possibly. The other thing that I like to tell people is that a raising tide raises all ships. If everyone who's in your match list is saying, well, I don't really have any matches, why would I want to do additional testing? Then there's never going to be more matches. So doing additional testing helps, it benefits everyone, both yourself and your matches. All right, let's see. We have the Olmsted subgroup chart for both Y-DNA and mtDNA. Is there a possibility for a similar chart for Family Finder for those that know Olmsted lineage? Family Finder is really tricky because it involves so heavily matches and shared centimorgans. And just like we don't list names with Y-DNA and mtDNA, we don't list names and projects for autosomal DNA. So that limits how it can be applied to group projects. I know there's been some talk about implementing a family connection like Ancestry has, but the difference is that Ancestry is primarily a records-based site, which means people are putting their genealogical information along with their names there and making that publicly available, whether it's to a match or not. So they're able to do a little bit other things with that. It's something we want to do, but again, there's limitations with it, unfortunately. All right, Project Y STR tables include self-reported elder ancestor, optional tester's last name and kit number, and Y haplogroup, which easily IDs big Y terminal SNPs. In many surname projects, there are project subgroups that are not related in same project. Will FTDNA give contact info for a kit number to callers? For example, ATDNA testers in a Y surname project who don't fund or facilitate big Y testing of their lineage. We wouldn't give out that information, but we could contact that person on your behalf and request that they contact you. That's also something, if you're in a project, that a project administrator could facilitate on your behalf. Will there be an age calculation between 2 to 5 individuals with same haplogroup in years? There's the TIP calculator, which gives you a time to most recent common ancestor estimate for matches. You can read more about the TIP calculator in our blog. I think I might not be fully understanding what your question is, though. So if you want to direct message me to clarify, I can try to work some of that out. And I know I'm saying to contact me directly, which I'm totally happy to, but if I don't get to you immediately, it's because I'm probably answering someone else, but I will get to you. So yeah, let me know if I can help more with that. And I recently got a big Y on a 10-year-old sample. So yeah, there you go. It's, it can vary a lot. Let's see, I don't recognize the little flag symbol. Is there a way to look up what it means? I was just looking on my phone, so I couldn't hover over it. I believe that when you hover over it, it tells you what the country is. But yeah, I don't know if it does that on mobile or not. I know sometimes things aren't the most mobile friendly, this being one of it, because the chart can stretch out so much. So I would recommend looking it up on a laptop. Let's see, I will be excited to provide info to your groups once we have our manual adjustment. Our STRs are wild with GD10 at 111 for 2 people with common ancestor, 1750 to 1800, and averaging 2 private variants. You can see how the wild STRs will mess up our dating as they already do on Discover now. Yeah, STRs, like I have some stories on some STRs. I came across one person who it was 2 brothers, and their they had a genetic distance. They were two full-blooded brothers, and they had a genetic distance of like 8 at the 111 marker, and they were born 40 years apart. And in between, the father had undergone like some chemo, and I think like lived near a nuclear reactor or something. And so it caused all sorts of mutations, because as men age, they start to develop more mutations like in sperm. So if you have two sons who are separated by many years, they are going to have a higher number of mutations. So yeah, some wild stuff can happen with that, which is why I recommend Big Y. It's a lot more stable. Oh, let's see. My second cousin has tested the Big Y and is RB many numbers. Our shared great-great-grandfather was an only child. Is it unlikely to get many matches? You can also, I mean, it depends, if the great-great-great-grandfather was very prolific, then you still would get a lot of matches, because, again, SNPs are fairly stable. And then also, if the child of that only child, had a lot of children, again, it could be a lot of ones. I'm always hesitant. A lot of times people will ask, is it worthwhile? Am I going to get a lot of matches? Am I going to get few matches? You don't know until you test. And sometimes you find out stuff like, maybe that great-great-grandfather wasn't who you thought it was. I'm not saying that that's the case with you, but people find out interesting things through testing. So I can't really say definitively if it's gonna help you or not. Sorry. Let's see. How do I save the question and answers in this chat? Well, the chat is being recorded, and so it should be available. I think it's getting recorded. Courtney, is this getting recorded?
This is getting recorded, yes.
Okay. Yeah. So it'll be available, and you'll be able to see my cat and pause and zoom up on her. That's really the main attraction of recording it. Let's see. Do you know of any upcoming sales on Big Y, particularly upgrades? We're having sales right now. Check out our virtual booth. We have a lot of sales going on. If there were no matches at 67, is it worth doing the Big Y? That pretty much got answered with the other questions that I answered. I have 2 accounts on YDNA for my brother and one for my maternal cousin. I just ordered mtDNA. Is there a tutorial site to help a beginner like me understand the results that come from this type of testing? Yes, our help center is a really great resource. And not to toot my own horn, but toot, I wrote some of it, so it's pretty great. And it's going to have a lot of information there. From Joy, thanks. You're welcome, Joy. How do we contact you directly? If you're talking about me directly, you can talk to me in the booth. I don't personally, I don't do a whole lot of customer facing work. I'm pretty busy with creating learning material in the help center, so my time is kind of stretched thin with doing that. But there are really great people in customer service, especially the Big Y specialists. They know as much, if not more, than I do when it comes to Big Y questions, and they're a really great resource. Our project subgroups always decided by admins? Yes. It looks like I probably have time for 1 or 2 more questions. So let's see, I tested 3 men from my great-grandfather's line, 2 from one son and 1 from another. Each one had a single different variant than the other 2, so they are all 2 variations off from each other. Is that common for people who are so closely related? I wouldn't say it's common, but it's not unheard of to have different siblings have different mutations. Bennett Greenspan and his son and his father all have different SNPs from each other, and that's just in 3 generations. So stuff like that happens. All right, so last question. Let's see. FTDNA should consider donation Big Y upgrades through group projects for kits where the tester or facilitator have died. You are absolutely right. And that is actually something that we are working on implementing in the very near future. We're going to make on the beneficiary section, we're also going to add a section where people can check that they are willing to have someone upgrade their test for them. So, that way if they pass on, or if even just financially they, it's like, you can test me if you want. I don't really care. I'm not going to pay for it. You can make it available for upgrades. So that is something we're pretty excited about. And unfortunately, that's about all the time that we have here. I know there's more questions. Like I said, I'm sorry, I tried accommodating as much time for questions as I could. Stop by the booth. We'll help you out as best we can. And to the person who asked me about the close-up of my cat, you can DM me and I can send you some cat pictures if you want. So, all right. Thank you. Thanks for attending. And I hope you all have a fantastic rest of the conference.